Iron, Part 3. This is going to look at biochemical functions. Now here's an interesting thing. Cytoplasmic iron may be toxic as it reacts with oxygen, forming the superoxide radical, which is going to be extremely toxic to all cellular materials, plant or animal. Therefore, we've got to have iron in. But iron can be problematic depending on where it's at. It will react with oxygen. That picks up oxygen and releases an O negative, which is the superoxide radical, which is very reactive, very destructive. Therefore, iron must be kept at low levels. But we need it, we've got to keep it at low levels just in case. In plastids, such as chloroplasts, iron is converted into iron 3 hydroxide, which is the building block of ferritin. Ferritin is stored until needed. When the reverse reaction occurs and the iron is released into the cell, the tendency of iron to form chelates and undergo valence changes is extremely important. Remember, iron does this fairly easily. Because of that, we must use chelates and other materials to move it around or to complex with it. We are going to start out with succinyl-CoA, which is a common material that is going to combine with glycine, adding iron, releasing off carbon dioxide and acetyl-CoA. This will yield gamma-aminolevulinic acid. That looks like a monster thing. It's got a carbon backbone. It's got the double bonded oxygen, which makes it an acid group. It's also got the amine from the glycine. If we take two of these, we combine them and we get a material called porphobilinogen. Porphobilinogen is the building block of tetrapyrrole. Put 4 of these together and we get this really unique looking molecule. Look at that. It's got the amino groups on the inside of these 5-sided rings. It has a very complex ring structure, which is really pretty neat. This, if you add iron to it, will give you cytochromes, cytochrome oxidase, catalase, peroxidase, and leg hemoglobin. Now, these, with the exception of the leg hemoglobin, are very important in animals as well. The leg hemoglobin is important in legumes because it is the material used by legumes to fix nitrogen. Very important material. This has all the carbons mapped out. The 4 nitrogens on the inside bind onto the iron, which would make this a ligand. This particular molecule is leg hemoglobin. We've got another possibility. We can add magnesium onto the inside, which gives us magnesium protoporphyrin. Out of that, we use iron to give us protochlorophyllide. Out of that, we make chlorophyll. Chlorophyll looks like this. This is part of chlorophyll A. We can change this around and get different types of chlorophylls. This is all the biochemical part, but you can see how important this particular process is. The tetrapyrrole can be used to make cytochrome, cytochrome oxidase, catalase, peroxidase, leg hemoglobin, chlorophyll A, chlorophyll B. Therefore, this is one of the building blocks of all sorts of biochemical stuff. This is how really important iron is in the physiology of organisms.